Hello and welcome to this video looking at how gene regulation is carried out post-transcription and then post-translation. We'll take a look first at post-transcription. So here we've got our region of DNA. And we've got this part, which is our transcription unit. This is the part of the DNA that we're interested in. So all of the DNA of the gene, both the introns and exons, are transcribed. So the introns are the non-coding regions, and the exons are the coding regions. So all of this is transcribed initially, and which is what we've got here. This is primary mRNA. But then this primary mRNA is then edited, and the RNA introns are removed, and the remaining mRNA exons are joined together. So some introns may themselves encode proteins, and some may become short, non-coding lengths of RNA involved in gene regulation. And some genes can be spliced in different ways. So how we get from primary mRNA to what we call here mature mRNA, and this is because this is containing only the exons, only the coding regions. It's done by splicing. And splicing is literally just like the removal of the introns from the mRNA. And that is catalyzed by the enzyme endonuclease. So we can also have what we call alternative splicing, which creates a lot of variation. So we could actually cut out this entire region so that it would become a different mature mRNA. So a length of DNA with its introns and exons can, according to how it's spliced, encode more than one protein. So if we had this, it might encode for one protein, but this might encode for a different one. So the mature mRNA is shorter, and then that is translated to produce a protein. Okay. So that's how gene regulation is done at post-transcription level through splicing of the mRNA. And this is like what happens post-transcription. But if we're looking at post-translation level of gene regulation, it involves the activation of proteins. So we've got quite what we might call a cascade of reactions. Depending on how many marks a question is worth, you might not need to go into every single little kind of individual reaction. You could just say that there's been a cascade reaction if it was only like 2 marks or something. But I'm going to go into the details for now. So we have, I'm going to do this, so we've got first here a signalling molecule such as protein hormone, glucagon, binds to the receptor on the plasma membrane of the target cell. So we've got it bind to the receptor. So this is what's happening here. So you remember it's a complementary shape, so this will be specific for one kind of molecule. This then activates a transmembrane protein. So we've got a transmembrane protein which then activates a G protein, and this is the G protein that's been activated. The activation of the gene protein then activates adenyl cyclase enzymes. They're here. Adenyl cyclase enzymes. And then the activated adenyl cyclase enzymes catalyzes the formation of many molecules of cyclic AMP from ATP. Cyclic AMP is an important second messenger involved in this activation. So we can see like ATP, we're going to have got rid of 2 phosphate groups, because adenine triphosphate, adenine monophosphate. So we have cyclic AMP, which then activates the pKa protein, which stands for protein kinase A. So we've got this activated, and the activated pKa catalyzes phosphorylation of various proteins, as you can see here, and it hydrolyzes ATP in the process. So we've got ATP to ADP, so one phosphate group is being released as like energy. So this phosphorylation activates many enzymes in the cytoplasm, for example, those that convert glycogen to glucose. So pKa may phosphorylate another protein, CREB, which is CAMP response element binding. So this is a CREB protein. And then this then enters the nucleus, as we can see here, it's entering the nucleus and act as a transcription factor. So as you can see, there's a cascade of reactions. So unless it was like quite a lengthy mark answer, I don't think you need to go into every single one. Transcription factor. To regulate gene transcription, which you can see here, the DNA. So this is like the turning of genes on and off. So a transcription factor, like we've said, is the protein or short non-coding RNA that can combine with a specific site on a length of DNA and inhibit or activate transcription of a gene. So it's important that we know that it can be turned on or off, okay? So this is how we've got regulation, post-transcription and post-translation. Thank you for watching.